The views and opinions of findings and/or devices discussed in this podcast are those of the host, subject matter experts, and/or guests. Facts represented constitute our understanding at the time of the podcast, whereas updated factual information may be developed. They should not be construed as pronouncing an official Department of Defense's position, policy, decision, or endorsement. The hosts and guests of CUBIST may be defense contract personnel who support TBI COE. Status of all hosts and guests will be identified during introductions to the podcast. Hi, welcome to the Clinical Updates in Brain Injury Science Today, or CUBIST, a podcast for healthcare providers about current research on traumatic brain injury, also known as TBI. This program is produced by the TBI Center of Excellence, or TBI COE. I'm your host, Dr. Katie Monty, a contractor in support of the TBI COE. Today I'll be speaking with Dr. Donald Marion, a neurosurgeon and TBI subject matter expert who is also a contractor in support of the TBI COE. Don and I will discuss a study titled Association of Blast Exposure in Military Breaching with Intestinal Permeability Blood Biomarkers Associated with Leaky Gut by Qingkun Liu and colleagues, and published in the International Journal of Molecular Sciences in March 2024. In addition, we are pleased to welcome Dr. Phil Karl to the podcast. Dr. Karl is a government service employee serving as a nutritional physiologist with the Military Nutrition Division at the U.S. Army Research Institute of Environmental Medicine. He is a subject matter expert in the gut microbiome, especially as related to military nutrition and performance. We will first hear a brief synopsis of the study from Dr. Marion, and then we will bring Dr. Karl into the discussion later to explain some of the concepts in the study specific to gut microbiota research. Don, thank you for being on the podcast and for bringing this article to our attention. Gut microbiota has become somewhat of a hot topic in medical research. I've been seeing more emerging evidence and its role in TBI and blast exposure. Can you share why this study was done?
Sure. Hi, Katie, and good to speak with you again. During the past few years, there has been an uptick in the Department of Defense concerns about the impact of low-level blasts, or blasts that are not severe enough to cause a concussion based on the DoD definition. However, it is becoming increasingly apparent that service members exposed to low-level blast, such as breachers or 50 cal operators, often complain of some of the behavioral and cognitive problems associated with post-concussion syndrome. Most have assumed that those symptoms are due to brain injuries caused by the frequent exposures to low-level blast. But recently, several investigators have identified leaky gut or intestinal permeability caused by low-level blast exposure as an alternative cause of the post-concussion symptoms because of the effect on the brain of the resultant blood-borne proteins and bacteria from the leaky gut. This study was done to further investigate the role those proteins and or bacteria may play in the behavior and cognitive deficits associated with low-level blast.
Thanks, Don. And can you tell us, what is leaky gut exactly? I've heard that term used quite a bit in the media and some articles I've read.
Sure, great question. Leaky gut refers to the disruption of the integrity of the physical barrier between the intestines and the bloodstream. Generally speaking, when there is a change in the composition of intestinal bacteria, or the microbiota, and the change is significant enough to result in its inability to function optimally, this is called microbial dysbiosis. That is, bacteria present in the intestines can leak out into the bloodstream. Under certain conditions, there also is the possibility that the connections, or tight junctions, between the cells that line the intestines can be disrupted by external forces. This condition also can result in abnormal intestinal permeability or leaky gut.
Thank you for clarifying that. So, Don, how was the study done?
Katie, the study was conducted during military breaching training between August 2016 and July 2018. The training involved heavy wall breaching exercises, and 33 active duty male breachers encountered at least 2 breaching charges where they were positioned in a breaching stack formation at a minimal distance of 40 feet from the blast source on a concrete wall. Blood samples were available for 30 participants. All participants met the military's physical requirement standards and were fit for duty. Blood samples were collected serially, pre-blast, that is in the morning between 7:30 and 9 a.m., and post-blast in the afternoon at 4:30 to 5:30 p.m. on the training day, and a third sample on the next day in the morning between 7:30 and 9. And the samples were stored at minus 80 degrees. On average, the post-blast time point was about 1 hour, and the follow-up time point was 16 hours following exposures to the blast. During the course of training, participants completed a 32-item paper and pencil health symptom inventory at pre, post, and follow-up blast exposure time points in conjunction with each blood draw. Items on the symptom survey were the same as those on the Rivermead post-concussion symptoms questionnaire, with additional items regarding concussion symptomatology, and each item was ranked using a 5-point Likert scale. Blood samples were collected in PAX-gene blood RNA tubes. RNA was extracted and globin mRNA removed. Total RNA sequencing libraries were prepared from RNA samples with RNA integrity numbers. Final libraries were then evaluated and sequenced.
Can you tell us about the intestinal permeability biomarkers that they examined?
Sure, Katie. They measured several intestinal permeability biomarkers, and they included zonulin, lipopolysaccharide binding protein, claudin-3, and intestinal fatty acid binding protein.
What were the results of the study?
The average age of the service members was 30 plus or minus 6.5 years, with 83.3% serving as combat engineers. Mean duration of service was 9 years. 18 individuals reported a prior history of mild traumatic brain injury. However, there was no association between history of mild traumatic brain injury and career breaching history, which ranged from 0 to 400 plus self-reported career breaching events. The majority, or 53%, reported 10 to 99 breaching events. Blood samples were collected from 30 of the 33 participants. Blood RNA was mapped to the human transcriptome. Blood RNA was mapped to the human transcript. Candidate microbial sequences were identified, and the relative prevalence of the top 3 microbial phyla found in the blood were characterized as proteobacteria, actinobacteria, and firmicutes. For a subset of participants, or 24 of the participants, changes in the intestinal permeability biomarkers zonulin, LBP, claudin-3, and intestinal fatty acid binding protein were assessed during training. Ultimately, data was only available for 21 service members and showed that both pre and post-blast, or 1 hour, and pre and 16 hours follow-up, changes in intestinal permeability biomarker levels were correlated with cumulative blast exposure. The service members reported headaches, concentration difficulties, dizziness, and taking longer to think acutely at 1 hour following blast, and these symptoms dissipated within 16 hours. For each symptom, participants were assigned to either of 2 groups based on changes in their self-reported symptoms, that is, increasing symptoms versus non-increasing symptoms, by comparing the symptom scales reported at pre versus post per subject. Then the increasing versus the non-increasing symptom groups were compared for differences in levels of intestinal permeability biomarkers separately across time points, and specifically post and pre. They found evidence for bacterial translocation into the circulation and alterations in intestinal permeability protein biomarkers 1 to 16 hours following blast exposure in the increasing symptom group. Zonulin levels were 47% higher, LBP protein levels were 12.5% higher, and intestinal FABP levels were 67% higher at 16 hours after exposure compared to pre-exposure levels. Claudin-3 levels, however, were 44% lower at 16 hours. Additionally, Katie, at 1 hour post-exposure, those reporting a headache went from 8 to 21 of 30 participants. Those reporting a longer time to think went from 4 to 15 of the 30. Those reporting dizziness went from 2 to 11 of the 30, and those with concentration difficulties went from 7 to 12 out of 30 service members. Because these symptoms were associated with changes in bacterial translocation and intestinal permeability, the authors suggested that intestinal permeability may be linked to decreased cognitive functioning. The authors claim that this is the first study that shows that exposures to blast in a military operational setting contributes to bacterial translocation and intestinal permeability, along with associated cognitive symptoms, and that this establishes a role of the gut-brain axis in blast-related sequelae.
So just to summarize for our listeners, they found a correlation among pre versus 1 hour post-exposure and pre versus 16-hour follow-up changes in intestinal permeability biomarker levels and cumulative blast exposure. And they also found an association between cognitive symptoms and changes in bacterial translocation and intestinal permeability. And that was determined based on levels of 4 circulating biomarkers.
That's great, Katie. You said it much more clearly than I could, I think, with all the detail.
So what were the limitations of the study?
There were several, Katie. First, the clinical symptoms such as dizziness or taking longer to think were self-reported, and there were no clinical assessments or objective measurements of these symptoms. Secondly, there were no independent control groups, and each breacher served as their own control by having a pre-exposure assessment. A third shortcoming was that no women were included. This bias is due to the predominantly male composition of the typical MOSs that involve breaching. But, Katie, I think the most important shortcoming of this paper is that the authors imply that intestinal permeability may have direct effects on the brain and, at least in part, be responsible for the cognitive and behavioral problems that were reported. But this study was not designed to demonstrate cause and effect, and showing a statistically significant association between cognitive or behavioral changes and elevation of intestinal permeability proteins or bacteria is certainly not sufficient to do that.
Thanks, Don. So what are the key takeaways for primary care providers in the military?
The study shows that low-level blast typically affects multiple organ systems, so the frequency and intensity of blast exposure probably should be limited as much as possible. When primary care providers evaluate a blast-exposed service member for complaints of post-concussion symptoms, they might also want to ask about eating habits, bloating, or other intestinal complaints.
Well, thank you for providing that excellent overview of this study, Don. I'd like to bring Dr. Phil Karl into the conversation now. Thank you for being on the podcast, Dr. Karl. Can you tell us why we should care about the gut microbiota in the general population and then more specifically in our service members?
Sure, Katie. To start, of all the cells in and on our body, at least half are microbial, and most of those microbes live inside of our gastrointestinal tract. What is more astounding is that those microbes contain hundreds of times more genes than us humans have in our genomes. So that enables the gut microbiota to provide functions that our human bodies do not have. Those functions include digesting foods that we cannot digest ourselves, producing vitamins and other small molecules that benefit our health, and stimulating our immune systems. In other words, the gut microbiota is generally providing health-promoting functions to our bodies. We, in turn, provide a hospitable environment for those microbes to live. It's really a win-win situation when working well, and our bodies and microbiota are constantly involved in a two-way conversation that maintains our overall health. However, that relationship can get knocked out of whack by stress to the human body or other factors like poor diet, which can cause changes in the intestinal environment. Those changes, in turn, can cause shifts in the microbiota that could contribute to some of the harmful effects of stress or poor diet on human health.
And do you mind telling us a bit more about these intestinal permeability biomarkers that we've been talking about and what your thoughts are on this study?
Sure. As Dr. Marion mentioned, the intestinal wall provides a physical barrier. It also provides an immune barrier between the microbe-dominated world inside the intestines and the inside of the human body. So if you think about it, the intestinal tract is really a hollow tube from the mouth to the anus. And if things don't pass through that tube into the human body, they just come out the other end. So the intestinal barrier is really providing a buffer between things that we don't want in our bodies and things that we do. So to do that, the barrier constantly monitors the environment and selectively allows certain compounds to pass from inside the intestines into our bodies while blocking compounds that we don't want into our bodies from entering. The selectivity of that barrier is controlled by multiple different mechanisms, and breaches of that barrier can be caused by dysfunction in any one of those particular mechanisms. So, for example, intestinal cells can be damaged, essentially leaving temporary gaps in the intestinal wall. Additionally, the machinery that controls the opening and closing of passageways between intestinal cells can stop working properly, leaving small gaps between cells. Dr. Marion mentioned tight junctions, and the tight junctions are what controls the kind of opening and closing of the passageways between intestinal cells. So essentially, the markers used in this study measure the different ways in which the intestinal barrier can become dysfunctional and lose the ability to effectively prevent harmful or other compounds from breaching the barrier and entering the bloodstream.
That's all very interesting. So the authors reported translocation of 3 microbial phyla that were found to be most prevalent in the bloodstream, as well as increases in circulating intestinal permeability biomarker levels. Is this enough to indicate microbiota dysbiosis or leaky gut?
That's a good and a tough question to answer. So I guess I'll start with the microbiota part. And I would say I don't think there's enough data here to really suggest any microbiota dysbiosis per se. And in large part, I say that because we are only looking at microbial RNA in the bloodstream. In order to make any claims of dysbiosis within the gut microbiota, we really need to obtain samples from inside of the intestines. Most researchers will rely on stool samples for that. I think one important consideration in this paper is also that we cannot be certain that the microbial RNA measured in the bloodstream is actually coming from the intestines. There could be other sources. Another consideration is that we're not looking at absolute amounts of bacterial RNA, but rather proportions. It could actually be that there's less total bacterial RNA in the bloodstream, just differences in proportions derived from different bacteria. So regarding leaky gut, there isn't really a threshold for any of these markers yet that could allow us to say that someone has a leaky gut. For example, these markers aren't like cholesterol tests where we know that levels above a certain threshold are high and would be associated with an increased risk of disease. We just don't have that knowledge yet for these biomarkers. That said, the observation that zonulin and LBP concentrations appear to be increased at follow-up compared to the pre-blast exposure are generally consistent with what we would expect with an increase in intestinal permeability. And I was really glad to see the authors examine correlations between changes in these markers of gut barrier function and symptomology, as that does help support their argument that the two are related. However, the time course of the symptoms doesn't completely match the time course of the gut barrier markers, and as Dr. Marion already mentioned, we cannot determine cause and effect from a study like this. However, the results of the study are nonetheless intriguing and important, as the microbiome gut-brain axis is an understudied but potentially important concept within military populations. And we just don't have a lot of information on this concept within these populations yet.
So the authors are reporting that 3 of the 4 intestinal permeability biomarkers had increased in circulation, and Claudin-3 levels decreased by 44% at the 16-hour follow-up. What do you think is the significance of that finding?
Yeah, that inconsistency does make the data a little bit harder to interpret. However, I think it might be related to the concept I mentioned earlier, which is that these markers are measuring different types of damage or dysfunction within the intestinal barrier. So Claudin-3 is a protein that makes up the tight junctions that we've been talking about. So these structures that help control the size of passageways in between intestinal cells. So the absence of an increase in Claudin-3 could suggest that the tight junction structure may not have been directly damaged by blast exposure. However, because intestinal permeability results from multiple pathways, not just a decrement in tight junction function, the absence of an increase in Claudin-3 does not necessarily mean that intestinal permeability was not increased.
Are there other factors that could have led to the increased biomarker levels and what the authors are calling bacterial translocation in the sample that may have not been accounted for?
There are. Again, important ideas to consider are that the bacterial RNA in the bloodstream may not have originated from the intestines, and we're looking at proportions of bacteria or RNA rather than absolute numbers. The rise in LBP might be a stronger indication than the RNA data of harmful bacterial components translocating into circulation. We do know, however, that diet can play a role in circulating LBP concentrations. So in response to your question, what the volunteers ate could have been relevant to the rise in LBP in addition to the blast exposure. Other factors influencing biomarker levels include things like sleep and physical activity. Both of these can impact intestinal permeability. Unfortunately, in this cohort, we just don't have much information on any of those factors.
And what is the natural day-to-day evolution of the gut microbiota in 30-year-old adult males without blast exposure? I've read that the lifespan of these bacteria is pretty short, and there's a constant turnover of the gut microbiota.
Yeah, you're exactly right, Katie. Unlike humans, bacteria will reproduce and grow at very fast rates. We'll do experiments in the lab, and you can measure bacterial growth over 2, 4, 8 hours. So it's very rapid. Over the course of the day in the human, we know that the composition of the gut microbiota and its metabolic activities are fluctuating in responses to normal growth dynamics of that community, but also changes in the intestinal environment. So as food passes through the gut, bacteria that eat that food will grow and multiply relatively quickly. They also respond to natural body rhythms. So you'll see changes in the microbial populations in response to just our natural daily circadian rhythms.
Could you share with our listeners some pearls of wisdom that you've learned from your research and education about maintaining a healthy gut?
Yeah, gut health and the gut microbiota are really hot topics these days for good reason. We're learning so much about the area. However, we are still in relatively early stages of uncovering all the ways that our gut microbiota may impact our health and how our own lifestyles can impact our gut microbiota. I'd say based on what we know today, eating a healthy diet full of fruits, vegetables, whole grains, healthy fats, and perhaps fermented foods appears to nourish and maintain a healthy gut microbiota. Getting adequate sleep and being physically active are also likely important. We do know that healthy diets, sufficient sleep, and being physically active are good for our overall health and our gut health. And we're beginning to understand that one reason may be because these behaviors benefit and keep healthy the community of beneficial microbes that already live inside of us.
Great. Thank you so much for being on the show, Dr. Karl. So that's all the time we have for today. You can stay up to date on future episodes by subscribing to CUBIST on iTunes, SoundCloud, Stitcher, or wherever you listen to podcasts, where you can also find links to the articles we discuss and other relevant resources. CUBIST is produced and edited by Vinnie White and was hosted today by me, Dr. Katie Monti. It is a product of the Traumatic Brain Injury Center of Excellence, a branch of the Research Portfolio Management Division under the Research and Engineering Directorate of the Defense Health Agency, led by Branch Chief Captain Scott Coda, Medical Corps, United States Navy. Thank you for listening to this episode.